So we've heard a bunch of great talks this afternoon about some of the most exciting, I think, conceptual areas of macroecology. And so what I'd like to do to wrap up is spend just a little bit of time talking about how we can move the field forward, not in terms of the questions that we're asking, but in terms of how we go about answering them. And in order to talk about that, I think it's useful to go back to the slide that Pablo stole from me last night, so I'm going through my laptop. And talk about the fact that back in the mid-70s, folks thinking about answering complex systems questions realized that some of the tools that had been used to really revolutionize areas of science had really focused on magnifying things so that we could see things in more detail. But that for emerging areas of research like ecology and economics, we might actually be better off finding a tool that would in some way filter out the details so that we could sort of understand these complex systems that would be overwhelming if we tried to sort of zoom in and focus on the individual particles. And the basic idea is just that we may be able to learn a lot about a system by stepping back to a distance and looking at the whole instead of focusing on the individual parts. And I think that Duraznet and a lot of the complex systems folks have been really insightful about how to study things like this. I think the real sort of test of the interestingness of a scientific idea is that it was anticipated years earlier by science fiction. And I certainly have a personal preference for Piers Anthony's 1969 description of the macroscope. Now, some folks in ecology don't, I think, understand or appreciate the value of a macroscopic approach as much as others do, and certainly I regularly encounter folks who sort of view this as the progression of ecology over the years, with the proud plant biologist carrying their quadrat frame going out into the field, and the noble insect biologist with their sweep net going out to sample, and the folks who have been talking this afternoon somewhere down here on the right. But I tend to think that these objections to macroecology really represent mostly a misunderstanding of the tool and what it can actually tell us. And I think that the talks we've heard this afternoon really sort of do an excellent job of justifying my personal perspective on the matter. So, I'm a big fan of macroscopes, I'm a big fan of macroecology, and so part of what I spend my time thinking about is how can we make these tools better? How can we make them better? And in particular, sort of how can we go from a sort of powerful but limited tool to something, well, a bit bigger. and you know, I think sort of things that I've learned over the years and sort of come to realize their own importance of, there are two ways that we can do this right now. And the first is to move from sort of focusing on analyses of a single data set at a time to trying to use the entire mass of accumulated ecological data that can be brought to bear on a particular question. This is something I started trying to do when I was a graduate student with Jim, and back then more data sets meant a couple of hundred sites compiled out of the literature. I think we're doing a little bit better than that now, and that's what I'm going to talk about here this afternoon, but we've still got a ways to go. And then the other thing is to move from focusing on a single pattern at a time to looking at multiple patterns simultaneously. Now, it's easy to justify why studying a single taxonomic group at a single site doesn't allow us to sort of say a lot about the general behavior of ecological systems. And so that's why, as macroecologists, we typically start with something like the breeding bird survey that has a lot more data associated with it. And we've got thousands of sites and hundreds of species, and we're feeling pretty good about ourselves at this point. But this is still just data on a single taxonomic group in a single region. It's a continent, but there's a bunch of other continents out there, and with a single very specific sampling regime. And so it's still a bit difficult to say whether the results that come out of an analysis of just BBS tell us something about the behavior of ecological systems in general. Fortunately, we're living in an era right now in ecology where we're transitioning from a phase where we're sort of largely limited by the availability of data to one where more and more data is publicly available, And now we're more limited by the speed with which we can acquire and analyze this data to answer questions. Which brings me to my sort of core philosophical point, which is that the more data that we throw at a problem now, the less time we have to spend later on going back to see if the results of our analyses are going to change when we look at a new taxonomic group, when we look at a new region, when we run into a different sort of sampling design. And so this is just sort of my most recent and I think best attempt to sort of use as much data as we can possibly get together. This is work done with Kate Tebow, who talked earlier, another student of Jim's, and my graduate student, Xiao Xiao. And I don't have time to sort of go into the details of the model, but we're basically testing state variable or maximum entropy models for the classic species abundance distribution. And the idea of these models is that instead of trying to model biological systems from the ground up, based on the details, we try to identify a couple of key constraints on the system, and in this case, that's the species richness of the system and the total abundance of all of the species in that community, and then figure out just what the most likely state of that system is, given those constraints. And in the interest of time, and knowing how incredibly smart everyone in this room is, I'm just going to channel Jeff West for a minute and say, obviously, this is the predictive form of the species abundance distribution, and move on to talking about how we went about evaluating it empirically. And we started with breeding bird survey data, which is great. A lot of great stuff done on BBS. We then added in data on the Christmas bird count. Now this is still birds. It's still North America, but not only do we get more sites and more species, but we get a very different sampling regime. And so if we see the same results between these two data sets, we can feel like maybe the details of how we sample those bird communities don't have too much of an impact on the question that we're interested in. We then added data on plants by using the forest inventory and analysis data set from the Forest Service, and butterflies from the North American butterfly count. And you know, we're doing pretty good at this point. We've got over 15,000 sites, over 1,000 species. We're hitting 3 major taxonomic groups, but we're still stuck in North America. And so in order to try to get a little bit more global, we brought in Alwyn Gentry's tree transects, which gives us some really good coverage, especially in the tropics. And then because I'd finally gotten tired of not being able to do individual level macroecology on mammals, and they're my background, my group collected a 1,000 community mammal database out of the literature. This is now available free of charge at Ecological Archives if you want to play around with it. And so we threw that in as well. And so that means as we're evaluating these models, we're working with data on almost 16,000 sites, almost 9,000 species on all 6 continents except Antarctica, and including 4 major taxonomic groups, including vertebrates, insects, and plants. And we can grab out a few example species abundance distributions, plotted here as classic rank abundance distributions, and we can look at how the observed form of the distribution, which are the circles in these graphs, line up with the theoretical prediction, which are the black lines, and things look pretty good, but we don't want to sit through 16,000 individual slides to see how well this is working. And so what we did was we took the predicted rank abundance, the predicted abundance for each rank at each site, and we plotted that against the observed or empirical data. And since we've got over 100,000 data points on each one of these 2 graphs, I've just color-coded them hot to cold so you can see where most of the overlapping points are. And what we can see is that for the birds, at least, we can actually do a very good job of capturing the form of the abundance distribution across sites by just using these simple models. So almost 90% of the variation. And if we look at the other studies as well, we can see largely the same thing for trees, 80 to 90% of the variation, over 80% for the mammals, and over 90% for the butterflies. And so if we sort of cut the hot areas out of each one of these graphs, and we overlay them onto a single plot, we can now, in a single graph, sort of synthesize the analysis of over 16,000 sites, 6 continents, 9,000 species, 4 major taxonomic groups, representing over 50 million individual organisms. And I think the advantage of sort of trying to take macroecology into this big data informatics age is that we can now be quite confident that the results of the analyses that we've conducted are going to be consistent. Even if we go out and we look at a new area, if we go out and we look at a new taxonomic group, we've sort of looked thoroughly enough that none of that is going to matter, and we can be confident that this is a jumping off point. That's not to say that these models are right, it's not to say the analyses that we conducted are right, but it is to say that this analysis is relatively done. We don't have to go back and revisit it over and over and over again before moving forward. So, that's sort of my one data set to many data sets spiel. I don't have time to go into the multiple patterns in detail, but I will say that it's quite important even in the case of this research that we're doing right now, which is that looking at these state variable models only on the species abundance distribution looks like they do very well. It looks like we've got sort of a solid foundation to build on. But when we force these models to predict multiple patterns simultaneously, we start to see important weaknesses that then need to go back and be addressed. And so with that, I just want to say a really heartfelt thanks to Jim. Like everyone else has said, this has been an amazing place to go to graduate school. I really appreciate being introduced to the sort of macroscopic worldview. And I should probably also thank him practically for helping me write this talk, because as with many things in my career, as I sit down to write a new paper or I sit down to write a new talk, I realize that in fact Jim has often been talking about these things for at least a decade, if not actually doing them himself. And I think that actually metabolic scaling theory and the work that they've done on it is a sort of great example of how to do both these things, right? They've been building models that make multiple predictions, and they've been testing those multiple predictions using multiple data sets. And I think it's a real model for us to follow going forward in the rest of macroecology. And with that, I just want to look at do one more thing very quickly, which is that Pablo and I have both sort of attributed early work on the macroscope to Durazne. But back here on Wikipedia again, it turns out that there's actually been some controversy in this area. And basically what this paragraph tells us is that Jim Brown is both Durazne and Piers Anthony. And then he realized that this concept would be so controversial in macroecology that he's been building a long-term plan by introducing it first through science fiction, then through philosophy of science, and then finally in 1989 really bringing it fully to bear in ecology. Thanks.